Thank you for the introduction and the invite. So before I get started in diving into all of this data, which has sort of taken on a new face in the last month or so, I need to define some of the terms that I'll be discussing in this presentation. And when I talk about the maternal-fetal interface, I am not a biologist that studies the changes that occur in the mother in the uterine wall during pregnancy, establishment of pregnancy. I'm a T-cell immunologist, so I can't give you much detail. But when I'm talking about the decidua in this talk, I'm talking about the mother side of the maternal-fetal interface, and when I'm talking about the placenta over here, I'll be talking about extrafetal tissue. So it's not the fetus itself, it's derived from the fetus, and it's on the fetal side of the maternal-fetal interface. And as far as the immune system goes in the maternal-fetal interface, pretty much everything we know is under the auspice of setting up fetal tolerance. So NK cells, T cells, dendritic cells have all been shown to promote a tolerogenic microenvironment in the maternal-fetal interface. And primarily this has been studied on the decidual side. Not a lot has been studied about the fetal immune system and how that is involved in tolerance, and in this case, how viral infection changes the microenvironment in both the decidua and the placenta to influence immune responses to the virus. So why are we interested in studying HCV infection in the maternal-fetal interface? Personally, because I think it's a really interesting immunologic question. About 4% of mothers who have hepatitis C infection, chronic hepatitis C infection, transmit or vertically transmit that infection to their child. And about a third of those cases actually happen in utero, which is a little surprising to me based on numbers. So people have estimated that as few as one HCV infectious virion can give you active infection. And the numbers range from 1 to 37 active HCV virions can cause infection. When you take a mother who has an average level of circulating HCV in her peripheral blood, there's been estimates that 10 to the 13 to 10 to the 14th virions actually can access the placenta during full-term gestation. So why then, if you only need one to cause infection and you're exposed to 10 to the 13th to 10 to the 14th, why is only 4% of these children getting transmission of HCV from their mom? And there's a couple pieces of evidence that suggest the fetal immune system is important in preventing this vertical transmission. The first of which is that one of the factors that's associated with vertical transmission is HLA genotype of the baby. And so it suggests that the T cells or other immune cells are involved in preventing this vertical transmission. And then, I don't see the pointer on here, the vertical transmission rate also is increased to about 20% in patients that are co-infected with HIV and HCV. So HCV and HIV both suppress the immune system, and when you have a co-infection with these two viruses, this immune system suppression is increased. So it suggests that the more you dampen the immune system, you increase the vertical transmission rate. So the hypothesis that we have for these studies is that the fetal immune response is critical for preventing vertical transmission of HCV. And the first clue we really had into this was from a paper from Hugo Rosen's lab, which was pretty elegant. And what Sylvia was able to document in this paper is, in this case, villus explants, so placental explants seated on matrigel. When she incubated these explants with HCV virions, not only were they able to take up the HCV virus, but they were able to respond to the HCV virus by inducing innate immune or molecules, ISGs, that have antiviral properties, suggesting the innate immune system was responding to this virus in the placental tissue. What about the adaptive immune system? As I mentioned, very little, if anything, is known about the adaptive immune system in the placenta and the decidua during chronic HCV infection. So we didn't really have a lot to go on, and we could sort of take some cues from what happens in the peripheral blood and the liver of patients infected with HCV. About 60% of people that are infected with HCV develop chronic infection. The other 40% develop acute infection. And acute infection is associated with an active T cell, CD4 and CD8 T cell response. And the patients that are chronically infected have alterations in the global innate immune and adaptive immune response. And when we look specifically at T cells, one of the hallmarks of HCV infection is that chronic HCV infection results in chronic exhaustion in these T cells. So normally during a viral infection, an acute infection where the virus is cleared, a naive T cell responds to the virus, proliferates into effector T cells that can then clear the host of the virus, and then they differentiate down the memory T cell lineage. In chronic infection, such as chronic HCV or chronic HIV, these effector T cells cannot clear the viral infection, and then they differentiate down a separate lineage of memory-like cells that are called exhausted T cells. And these T cells have decreased self-renewal, decreased effector function, and increased expression of inhibitory markers such as PD-1, TIM3, TIGIT, and others. And so we wanted to examine at the maternal-fetal interface, are HCV-specific cells more exhausted-like, more memory-like? What is their phenotype, and what is their T-cell repertoire? So our first hurdle was to actually identify T cells in the maternal-fetal interface. And hopefully you can all see this, but in the top here, I don't have, the pointer's not working, but on the left is the maternal-fetal interface from an uninfected mother and child, and on the right is an HCV-infected maternal-fetal interface. And I'm showing you T cells in the white, PD-L1 and inhibitory marker in red, and DAPI to stain the nuclei in blue. And what we have been able to see is that at the maternal-fetal interface in HCV infection, there's an increase in the frequency of T cells. And when we look at the placenta, we see the same thing. And the arrows point to the T cells. This is an HCV-infected placenta, and you can see there are T cells. There are, in fact, T cells in this microenvironment in HCV infection. And the frequency of these T cells is increased in the placenta by immunohistochemistry compared to an uninfected control placenta. Now, immunohistochemistry is great for localizing cells, but what it's really difficult to do with this type of analysis is understand what the phenotype of these cells are. So Christine Hurtado, working with Hugo Rosen, was able to use flow cytometry to identify phenotypic differences in T cells at the placenta and decidua from control in the shaded or the black symbols, and HCV infected in the unfilled symbols from the placenta, decidua, and the cord blood. And what she found was that in the placenta and the decidua, the T cell population is enriched for effector memory T cells in both the control and the HCV infected. And that's not really surprising because we know that in peripheral tissues compared to the peripheral blood or other lymphoid organs such as the lymph nodes, these tissues are enriched for resident memory cells, virtual memory cells, effector memory cells. So that wasn't surprising. It was kind of surprising to us, though, that there really was no difference between control and HCV infected. So we wanted to see if, despite a lack of phenotypic difference, whether there was a functional difference between HCV infection and control T cells in the placenta and the decidua. So what we did is we took out cells from the decidua and the placenta, stimulated them with a polyclonal stimulator, PMA and ionomycin, for 4 hours in the presence of Burchill A, and looked for cytokine or functional responses from these cells. And what we found that in the red, the HCV-infected cells produced much more interferon gamma in both the decidua and placenta compared to the control placenta and decidua, suggesting that HCV infection at the maternal-fetal interface maybe is skewing the immune response from a tolerogenic Th2 type of cytokine profile from the T cells to a more antiviral Th1 profile from these T cells. But this is global T cell populations, so we can really only take limited interpretations from this. And our real question is, are there HCV-specific T cells at the maternal-fetal interface? Because we want to see how these cells, and if these cells are involved in preventing vertical transmission of HCV. To do this, we took advantage of MAC tetramer technology to use flow cytometry to identify HCV-specific T cells themselves. So in the upper left-hand corner here is an example of a flow cytometry plot looking at CD8 T cells that bind to HCV tetramer or are HCV-specific. And it's pretty obvious that this is not that impressive. I mean, 0.046 or 7% is not impressive. And this is a problem we run into with HCV T cell responses in the peripheral blood, as well as the placenta and the decidua. So since nobody has ever really reported HCV-specific T cells at this interface, we felt we needed to go a step further. And so we utilized a technology or a technique called tetramer enrichment. And this was pioneered here in Colorado with Pippa Merrick, John Kapler, Ross Kettle, and at the University of Minnesota with Mark Jenkins and Steve Jamieson. And what we can do is we can enrich for HCV, in this case specific T cells, using Miltenyi bead separation. And what people have shown in some of the publications, many people have published it, some of them are listed here on the left, is that this technique is highly specific, there's very little if any background, and is able to pull out virtually every single antigen-specific cell. So it's perfect for understanding T cell responses at low frequencies like we have here in the HCV-specific response at the maternal-fetal interface. So using this technique, what we wanted to do is we wanted to look at the phenotype of these cells, what their affinity for HCV peptide is, and what is their TCR repertoire. And that's what I'll be talking to you today is the TCR repertoire. And before we get into the actual data, I thought I'd give as simplistic of a view of the TCR repertoire as I can possibly give. And T cells have a T cell receptor, an antigen receptor that's composed of an alpha chain and a beta chain. There are many different genes that are part of the TCR or alpha or beta locus. During recombination, these receptors can be combined, and it's estimated that humans have potentially 10 to the 18th T cell receptors in their immune system. They have the potential to form that many. But we know that an average human has about 1 times 10 to the 13th T cells in their body. So it's clear that we don't all have 10 to the 18th circulating TCRs. And so people have back calculated that the average person has about 25 million different T cell clones in their body at any given time, expressing different sequences in their T cell receptor. That's what gives us our diversity to respond to many different pathogens that evolutionarily we had never seen as a species until recently. And one of the fundamental concepts of T cell biology is clonal selection during infection. And here's a representation of some data from Jeff Friedlinger's lab. I think he was at North Carolina at this point. What happens to the repertoire, in this case, this is LCMV-specific V alpha repertoire prior to infection and post-infection. And what happens in a naive repertoire, you can see this is a pie chart representing all V alphas that are utilized by LCMV-specific cells prior to infection. Following infection, this diversity gets less and less and less, such that you have clonal selection, and this orange piece of the pie there is most likely the most fit cell that is able to ward off secondary infections. So what we wanted to ask is the TCR repertoire from HCV-specific cells different between the decidua, or the mom side, and the placenta, the baby side? Because what's the setup in this microenvironment is that the mom has chronic infection and cannot clear the infection, but the baby doesn't develop productive infection. So potentially the TCR repertoire is more effector-like and more diverse in the placenta, whereas in the mom is more clonal. And to do this, we utilized a recently available technique from 10x Genomics. So TCR sequencing is not new, but we utilize the 10x Genomics platform. And what we can do is we can isolate HCV-specific cells and identify the T-cell receptor sequence in each one of these cells individually, so we can get a look at the T-cell clones in both the mother and the baby. So it can tell us if there are any alterations in the V-alpha or V-beta gene usage. And so our first attempt at this, what we found on the top is the TCR alpha and beta chain usage. This is just V-alpha. This isn't diving deep into the sequencing from both the placenta on the left and the decidua, the mom, on the right. And in the bottom is the pie chart representing the different alpha chains, the V regions used in the placenta as well as in the decidua. And what you can see from the pie chart is that there's a much more diverse repertoire of HCV-specific cells in the baby than there is in the mother. The mother is essentially, the T cells in the decidua are essentially clonal as far as V-alpha usage. They have different V-betas, but they use one V-alpha. So it suggests to us that maybe, well, first off, the repertoires are completely different between the two groups in this microenvironment, that the placenta T cells are different than the decidual T cells. And since they're diverse, it's possible that the reason that you get low levels of vertical transmission is that they can respond to different epitopes with higher affinity, and which we'll address in the future. So what I've shown you so far is that HCV infection results in the accumulation of effector cells that produce functional Th1 cytokines at the maternal-fetal interface. Hopefully we're getting past the point where we can say conclusively that there are HCV-specific cells, T cells, at the maternal-fetal interface, and the TCR repertoire of HCV-specific T cells is really non-overlapping and strikingly different between the placenta and the decidua. So where we want to go with this is a couple of the questions that we have are the T cells, the HCV-specific T cells from the maternal-fetal interface derived from the mother or from the child? Are they crossing them from the mother, the maternal-fetal interface, into the placenta, or are they coming from the placenta, or the fetus, excuse me? And then we want to see if this difference in repertoire correlates to a difference in affinity to respond to HCV peptides, because the affinity for HCV peptides or peptides in general is important for effector function. So if they're much more highly avid, they may have a much more ability to respond to these HCV peptides in this microenvironment. And then the ultimate question, which we're trying to answer, is are these really critical for prevention of vertical transmission? And with that, I'd like to thank the people that were involved in this. This has really sort of been a long, drawn-out project with a lot of people contributing in the Rosen Lab: Christine Hurtado, Lucy Golden-Mason, Rachel, Sylvia, Mel, who's no longer here, Lin Ling. The immunofluorescence was done by Jeff in Beth Tamburini's lab. We had some help from the genomics and core for the TCR sequencing, and Kent for helping us with that, and the RNA bioscience initiatives. We got specimens from the UCD pathology core as well as Michigan State University. And then, since I have a little bit of a time, I'd like to put a plug for a talk that's later today, a Gallup seminar. It's another collaboration between myself and the Tamburini lab, and Beth will be talking about our data involving dysregulation of lymphoangiogenesis and how that promotes fibrosis in chronic liver disease. So with that, I'll be happy to take any questions. In the thymus of each. Well, it depends on age, right? Yeah, I don't know. Certainly, there's, the fetal thymus isn't set up to produce T cells until later. A lot of the hematopoietic cells come from the fetal liver. I don't know at what stage this T cell difference is set up. If it's set up early, I mean, these are full term, so we're taking a snapshot after delivery. So we don't know if, these T cells, well, first off, we don't know if they're coming from the mother or the baby, but we don't know if the T cells are involved from the very first step to prevent vertical transmission. I don't think that answered your question, but I guess I don't know.
I don't know.
Thanks. So as far as things like NK cells in the decidua, those have been, NK cells and dendritic cells have shown to be more immunoregulatory. There's a lot of regulatory T cells in there that there are your traditional FOXP3 positive Tregs, but there are other CD8 T cells. It seems like they're more immunoregulatory. Nobody knows anything about their antigen specificity, whether they're specific for self-antigens and suppressing cells that come in who are trying to reject the fetus. It's a tolerogenic microenvironment. Whether the repertoire of T cell receptors in non-HCV is unknown, if that could be. I don't know. A lot of the studies that are looking at vertical transmission are done in rodent models, and the setup there is quite different. It's easier to manipulate and understand the factors that are involved. There is some association with HCV or other viral infections and termination of the pregnancy. So it would make sense that if you decrease this microenvironment and make it less tolerogenic, you make it more inflammatory, that some of those fetal-specific cells that are coming in to rid the body of the fetus would then cause abortion of the termination of the pregnancy. So there are, I think that you're sort of on the right path, that this changing of the microenvironment is more, since it's less tolerogenic, is more permissive to fetal rejection. But that's not really that well established.
To build on that, on the very first slide, you say
right, from 4 to 19.
Why is that?
I don't know that anybody really knows. I mean, I think this is a rapidly expanding field where there's almost no literature. So my suggestion would be, HCV chronically exhausts primarily the CD8 T cells. HIV knocks out your CD4 T cells. It's a double hit on the immune system. They also have indirect effect or direct effects on the innate immune system as well. So you are with both infections, you're suppressing the innate and the adaptive immune response. And now when you suppress both, you're more permissive to vertical transmission, because that to me suggests that there is a role for the immune system, whether it be innate or adaptive, in preventing this vertical transmission. Thanks.
Thank you very much.